Hi, I'm Prisha, and today I'll be reflecting on an article from Science Daily titled A MicroRNA Solves an Evolutionary Mystery of Butterfly and Moth Wing Coloration. I chose this article because it connects closely to our week 4 and 5 topics, which is non coding RNAs, genetic variation, and gene prediction. And I found it fascinating how something so small like a microRNA could control such a visible trait. To give a quick review, butterflies and moths have genetic variation in wing coloration due to the presence or absence of melanin, which give two types of variant, dark and light. For nearly 20 years, scientists believed that the gene cortex was responsible for dark or melanic wing patterns in butterflies and moths. That's because genomic studies kept mapping this trait to a region called the cortex. The assumption was that cortex must be the gene responsible for turning melanic coloration on and off. However, research at National University of Singapore, working with teams in Japan and US, found something so surprising. It wasn't the cortex at all. The true switch is a microRNA called MIR193, which sits right next to the cortex in the same region. When MIR193 was disrupted, butterflies lost their black pigmentation completely. However, before we dive further, miRNAs are actually small RNA molecules that play crucial roles in gene regulation, even such as suppressing the expression of target genes and blocking translation of selective mRNAs. And to gather evidence, the team used CRISPR-Cas9 to disrupt the MIR193 gene in three different butterfly species. So two were dark and black wing color, and one was a white-winged color butterfly. In every case, when MIR193 was disrupted, the butterflies lost their dark pigmentation completely. By contrast, when they disrupted the cortex itself and even three other protein coding genes in the same region, there was no effect on color. That was a strong evidence that MIR193, not the cortex, is the real effector. So, how does it work? MIR193 comes from a longer non-protein coding RNA called ivory. This microRNA directly represses the pigmentation genes, preventing melanin production. In other words, the presence or absence of this tiny RNA decides whether butterflies can grow dark or light wing colors. It was also found that the sequence of miRNA 193 is deeply conserved across most Lepidoptera species. And to test if this conservation corresponds to functional similarity, the researchers also tested in Drosophila flies and found that MIR193 also controls melanic coloration in these flies. Thus, its role in pigmentation is ancient and shared. This discovery overturns 2 decades of assumption. It shows that non-coding RNAs can drive major traits and genomic association may not identify the right candidate unless regulatory non-coding elements are also considered. It also shows that deep conservation across species shows that some regulatory mechanisms are ancient. So to reflect and relate back to Gen 2052, non-coding RNAs like MIR193 are powerful regulators of phenotype. From Gen 2052, we know miRNA causes transcriptional silencing, which is applicable to the miRNA role in melanin pigmentation, according to the article. In addition, a regulatory non-coding element causes visible genomic variation in wing color throughout the species. It goes to show that gene prediction requires looking beyond coding regions, as in this article the original assumption was wrong because only the coding genes were considered. And lastly, the discovery that MIR193 controls pigmentation in butterflies and also Drosophila fly is a perfect example of the principle learned in Gen 2052, where conservation across genomes points to an ancient and essential role. So that's all from me. Thank you.